world. This is truly a global conversation. Just to give you an idea, our panelists are among them, one of them in Melbourne, Australia, it's 2 a.m. in the morning. The other one is on the west coast of the USA. It's 9 a.m. in the morning, different days. But they're here to tell us about their field of expertise. This is truly a global conversation. We've had interest from around the world. Many will follow the video that we will post, and from which we will make shorter clips around the very many, around the complex and varied areas of immunology that we'll discuss today. I've mentioned what time zones we have represented here, also among registrants. We have folks from Cambodia, from India, from all regions of sub-Saharan Africa, and also interest from Latin America, because it's a global pandemic and because our interest in this field has to do with how all of this, the COVID-19 pandemic, affects the globe, how vaccine development is important for the globe. And of course, many of us have very specific ways in which the pandemic affects our lives in the countries, in the cities where we live. But it's a global conversation because it's a global issue. So since the beginning of, I should say, I'm Ida Jooste. I'm with the health team at Internews. What does Internews do? We walk the walk with journalists. And in this case, since the start of COVID-19, for many and most journalists around the world, it's been a plunge into health and science journalism. We've interacted with hundreds of journalists in this time and have walked the walk with them, as I've said. We saw how journalists had to be in step with virology, with immunology, with understanding the significance of clinical trials, and of course also misinformation. So I think one of the recent insights that we've had, we've also in this time written a very widely popular course called Let's Talk Vaccines, which has a section on immunology. And recently we've started to reflect on, how journalists have fared. What are the questions that remain for them, that remain complex for their lives as reporters, health and science reporters. Some have reported on these issues for decades or for many years, and some have been plunged, as I say, into health and science reporting through the pandemic. So what we've come to realize is when it comes to the section of immunology in our Let's Talk Vaccines course, and also in conversations with journalists, and the very many webinars and very many other interactions we have with journalists, is that this one area of immunology remains truly complex. So we thought we needed to unpack it. And so I just wanted to know how this has come about, because when we talk amongst ourselves and when we listen, we realize this is an area that needs to be unpacked. So at Internews in this time, we've also been very fortunate to form partnerships, and one of them is with the Sabin Vaccine Institute. So they're our partner with this event, and I want to thank them very much. I also want to thank the scientists, as I say, all the way from Melbourne in Australia to Nashville, Tennessee, to California at La Jolla. And they are the ones who are represented here on the call. So Christoph, can you go to the slide for us, please? The first slide. Thank you. So here are our three scientists. I'm going to turn my camera off so that you can see them properly. And of course, they will be interacting and helping us unpack this subject of immunology. On the left is Dr. Jennifer Juno. She is in Melbourne, Australia, as I have said. She's with the Peter Doherty Institute for Infection and Immunity. Thank you and welcome. Thank you again so much for being up at 2 in the morning for this discussion, Dr. Juno. Then we have Dr. Jim Crow. He's with the Vanderbilt Vaccine Center, that's in Nashville, Tennessee. I've seen some wonderful TED Talks by Dr. Crow. I really encourage him to watch them. In fact, all three immunologists on the line today were selected because they are brilliant at their fields, and also they're brilliant at communicating their science. So thanks so much, Dr. Crow, for being here. And then we have Professor Erica Sapphire. She's with La Jolla, which is, of course, in San Diego, in California. And she is with the Institute for Immunology's Center for Infectious Disease and Vaccine Research. And she's also the director of the Coronavirus Immunotherapy Consortium there. So she'll be telling us much more about our work, of course. And let's go to the next slide, because I'd like to take you through the format we'd like to follow for today. And what we actually have is, first of all, we thought we ought to listen in to the conversations that scientists might have amongst themselves when they ask each other about their fields of expertise. But also at the same time, ask each of them to consider that we on the call are journalists, and we need to write about these things that, as I've said, have been really new to all of us. So what we'll have is Dr. Juno will ask a question of Dr. Crow, which he will then answer. And then Dr. Crow passes on to Dr. Sapphire. Our first intention is just to find out what each of the scientists on the line specializes in, what interests them, what fascinates them about their field. And then, of course, Dr. Sapphire to Dr. Juno, asking her also about her field of expertise. Then Christoph, go to the last slide before we plunge right into Unpack Immunity, just to say that that's the very first thing that will happen, from one scientist to the next. And then, of course, all of you scientists on the line probably have questions for each other, questions which will keenly listen into, we being journalists. We will keep on prompting and prodding you to put this in language that our audiences would relate to. And then as the final section is, of course, where we journalists ask you the questions. It can be questions in science, questions that of issues in immunology that have been in the news, or follow-ups from anything that you may have mentioned. So that's over now to Dr. Juno. We can take the slide off. I'm asking Dr. Crow to kick us off about this in terms of this area of science. Thank you.
Great, thank you so much. It's a pleasure to be here and to have the chance to talk to everyone. So I guess to kick things off, we sort of need to start with the basics. So I'm hoping, Jim, that you can walk us through some of the basic steps of how the immune system functions to respond to a new virus that it's seeing for the first time, like SARS CoV-2. And then maybe we can move on to talking a little bit about how vaccines can be used to educate the immune system.
Yeah, thanks. That's already a complicated scenario. So I think when I think of the immune system, a lot of people use that word. Lay people say my system's run down, or I'm going to eat well so my system builds up. And they mean their whole body, but really the circulating blood, the blood vessels go everywhere in your body and all the tissues. And the components of the blood are really two main things. One are the red blood cells that carry oxygen, and you have to have oxygen for your tissues to live. But the other part of the blood is called the white blood cells, and that's the immune system. So it's in your blood, it fills your blood, and those white blood cells are really a major component of the immune system. So they flow everywhere, but they also sort of get off the train, and they stop in areas of your skin or your lung or your gut or whatever tissue is of interest. And so they circulate, and then the white blood cells will exit the blood and stay in the tissues. These are tissue resident or tissue local cells. And so that's where all the action usually happens. So viruses or bacteria, they come in through the surface, like through your skin, your nose, your mouth, various surfaces, could be your eye. And at that point, the immune system has to be out on the frontier, always looking and watching for things that are foreign and danger signals. So sometimes you have microorganisms on you, and it's normal. Your skin has microorganisms all the time. But if you have a cut and they get inside the tissue and start causing damage to your cells, then you have a second signal. So you have a foreign material and a second signal, which is damage. And parts of your immune system sort of light up and they say, wow, I see something that's not me. It's foreign material and danger is going on. And you have some general senses, and everyone knows when you have an infection, you have, if you use the Latin rubor, you have redness, tumor, swelling, dolor is pain, and calor is heat. So all of these are signs that the immune system is starting to fire off and something's going on. And so then the immune system has to figure out what is this material, and it usually sort of eats pieces of it. There are cells called phagocytes, phago meaning eat. So there's cells that eat, chop up the material, and then talk to nearby cells. And that process is called antigen presentation, antigen presenting cells. And so they pass along the information to the innate immune system, which just has to look out for all types of foreign things. And then that innate immune system calls in the troops, which are the system that can adapt. And we call that an adaptive immune system. So now you have all sorts of cells coming in, T and B cells that we'll talk about a lot more. And they're really the cells that do the job. So there's sort of surveillance, signaling of danger, activating, and then calling in other immune cells. So that's the sequence of what happens, I think.
I think we're going to dive a little bit more into that sort of adaptive side of the immune system. I think that's the area that each of us are particularly interested in. You've mentioned T cells and B cells. What are the jobs of each of those cells, and why are they different?
Well, the whole idea of adaptive immunity is that you've seen something once, and now when it comes back, you can respond faster and better with a stronger immune response. And so we've used terminology that we sort of stole from the brain people, and we talk about memory. So you might have an idea from childhood, and as an adult you remember it, or a taste. And when you taste that food again, your body has a reaction, you have a memory. The immune system's like that too. If you've seen a vaccine or an infection before, and now you're exposed again, you activate faster, stronger, and longer, actually. And so sometimes we call that boosting. The second exposure boosts you higher and faster. That's what we mean by adaptation. You're better the second time than the first time. And the system that really does that, there are hundreds of types of cells and subsets, and people like us, depends on, are you a lumper or a splitter? You can split these things into thousands of little nuances, but fundamentally you have what are called B cells and T cells. And B cells are the cells that make a protein called antibodies, and antibodies float around and they'll recognize and inactivate viruses and bacteria. And then T cells move around. They actually work as cells, and they find points of danger like a tissue that's infected, and those T cells will process, they'll speak to other elements, communicate to other elements of the immune system. And some of those cells actually kill the infected cells. They just take care of the cell, which is really a factory for more viruses. Some of the T cells will take care of that. So you have these proteins made by B cells, and then you have cells cruising around called T cells. And they actually also talk to each other. They're very cooperative. I think that's something that's very difficult to understand, but this entire spider web of immune cells, the ones that detect, that eat, that present, the ones that secrete protein, the one that kills cells, they're all interconnected by signals. They secrete things, or we call soluble factors, or things that go out into the liquid space and travel to another cell. They do that, but they also touch each other. They have what are called contact factors. So you have one cell will touch something on the other side, and we usually call that a receptor. So a receptor and a ligand will talk. And so cell A will tell cell B, I see danger, you should activate and get going and do your job here. So there's soluble factors that work from afar, can circulate even your whole body. And there's also very intimate contact. So I think that's the T and B cell and the jobs they do.
Yep. And in terms of the COVID-19 pandemic, what has really been the focus of your particular research and your interest in trying to understand the immune response to COVID?
Well, the fundamental way that most vaccines work, or the memory from prior infection that protects you, principally are antibodies. And these antibodies float around and they're waiting for the virus like COVID to come back. And when they come in, the antibodies will recognize the virus, cover it, and basically inactivate it in some ways. And antibodies have a lot of tricks. They not only do it just by blocking things, but they also have functions that they can bring to bear besides just being present or blocking. And so our laboratory has tried to harness, the natural good immune response that you make for yourself. We can turn that into a drug. So we look through the blood cells of people who've survived. We find an individual B cell that's making an antibody for COVID, pull that cell out, And through modern molecular biology, we can get the gene for that antibody. And that's the recipe, the letters of the code that specify the protein. And we can use that code to make the antibody in a factory. And so the antibody you were making in your body, we can remake it in a factory. And then we can give it to someone who's never seen COVID or can't respond to a vaccine. There's people with cancer or people who don't even have an immune system. And it's fantastic because we can move the immunity from one person to another person and confer instant immunity on the person receiving that antibody drug. And we can also use the antibodies to treat people. If they're infected, we just put the antibody in and the antibody has a beneficial treatment effect. So we've been discovering and developing antibodies to prevent and treat infection as drugs, harnessing the immune system for good.
The use of those antibodies has been, really interesting and really effective. But I guess variants have changed the equation a little bit, particularly with Omicron. So how have the changes in the virus impacted our ability to use those antibody drugs for prevention or treatment?
Well, the type of antibody that's used for drugs, they're basically two kinds. You can take all of the antibodies from one person, sort of bleed them a large volume and separate out the antibody containing liquid, and that's called plasma. So convalescent, meaning recovered from infection plasma, you can take one person's plasma, put it in another person, and you're just transferring everything in that person's blood from one to another. That kind of works sometimes. It didn't work very well. But the technologies we're using now are called monoclonal antibodies, where we make that one antibody from one B cell from one person. And that's great because they're very specific. They recognize exactly one place on the virus, and we know how they work. And that's great because for the FDA or EMA or other regulatory bodies, we can say, we know how this works. We know what is called the mechanism of action. Now, the problem is they're so specific. If the virus escapes from that place, if it changes itself and has a mutation, that antibody may no longer work. And that's actually happened to several of the drugs that were approved. It hasn't happened to all of them. The ones we made actually still have activity, and we'll see because the variants keep occurring. But then it's also possible to make new ones. So one of the companies' antibodies stopped working and they made a second generation. So the variants are a problem with COVID because so many people are infected. You have the opportunity for the virus to try out lots of mutations, and some of those will escape these drugs.
Fantastic. I think that gives us a really good introduction and a really good start to talking about a lot of the components of the immune response. I think we're going to switch now, and I think we'll give you a chance to interview Dr. Sapphire and ask her some questions about her work.
Yeah, thanks so much. Erica, welcome to the conversation. I'm glad you're here. I'm interested in your thoughts on how do you talk about vaccines versus antibodies? They both confer immunity, but they're very different. And I think some people get those confused sometimes. How do you think about that?
Yeah, exactly. People confuse those two things. So what a vaccine is designed to do is to show your immune system what the pathogen looks like. So the analogy I like to use is an Old West wanted poster, where it's a photograph of the bad guy in the post office so that everybody knows what to look for. A vaccine gives you a harmless piece of the virus. It's the key piece. So in this case, it's that surface spike protein of the coronavirus. It's what the virus uses to enter your cells. The vaccine just gives you that one piece. So it's not harmful in any way, but it shows your immune system what to be prepared for. So your immune system sees it, it recognizes it as foreign, can build antibody and T cell responses against that, and have some circulating as plasma and some held as memory. So that if you're actually infected, you don't need all that time to mount a response. It can clear it right away. So you're less likely to get sick. An antibody is what your immune system makes in response to the virus, like you and Jennifer have just been talking about. So you can give a vaccine to prevent infection, or you can give an antibody to treat infection or also to prevent infection. So the antibody is actually the component of your immune response. The antibody is a human molecule. It's what you make. And you can give it as a drug. So that's important because not everybody can be vaccinated or has been vaccinated, or we were vaccinated with an earlier strain of the virus, and maybe we are susceptible to something different that may have emerged. Maybe someone's elderly and vulnerable. Maybe they are being treated for a cancer in some way that depletes B cells. Well, the remarkable thing, the technology like Jim's lab and others do, is they can look in that whole repertoire of different antibodies you make. And the immune system can make, I think, a quintillion possible antibodies. So that's one with 18 zeros after it. And that means that each antibody is quite individual. They're shaped like a Y. They have two arms and they have a base. And at the end of the arms, there are variable loops that give you fingerprint-like recognition for a particular place on the virus. And so you'd have antibodies against this spot and spike, antibodies against this spot, this spot, this spot, antibodies against measles, antibodies against polio, anything you've ever been infected or vaccinated with. Among those quintillion, there will be some that are extraordinary. There are some that are duds, and there's a lot in between. And so the goal of an antibody therapy would be to look across that whole range of all the antibodies that a survivor has made or vaccinate has made, and you pick out the very, very best. So you pick out the needles in the haystack. You pick out the very best 1, 2, or 3 of that incredible range, and you grow them in large scale, and you can deliver them as a drug so that that person has the immune response of a rock star on board as soon as that treatment has gotten into their body. And so you can use that in a couple different ways. You can use that if someone's gotten infected and you want to be able to knock down the amount of virus to give their immune system a fighting chance. You can deliver the antibody after they've been infected. You can also deliver the antibody before they get infected. And so this is really important if we're expecting seasonal return of coronaviruses like winter cold flu and COVID season. If you know someone's vulnerable because they're elderly, they have underlying conditions, they live in a nursing home, they're a cancer patient, you can deliver them this antibody as a prevention, and it'll last, many weeks to several months to help them ride through so that they have the antibodies they wish their immune system had been able to make. So there are two different molecules. The vaccine is a piece of the virus that you show your immune system so your immune system can make its own response. And an antibody is something that'll help your immune system. It's what an antibody treatment is the antibodies you wish you had on board, but we're giving you the very, very best that somebody had made in a much greater quantity than you might make yourself.
Yeah, that's great. I'm intrigued by the audience that we have today all over the world. And COVID was or has been an international event, obviously. And you've been a leader in pulling together consortia. I know a lot of scientists sort of work in a little, almost in a closet and do their own thing. That's been the history of science, but you've been a leader in building consortia and running them. I wonder if you could explain, how that's worked in COVID.
Yeah. So the immune system is complex. Antibodies work in a lot of different ways. Some of them will mechanically inactivate that virus. Some of them, their job is to inspire the rest of your immune cells to come in and destroy the virus or clear the infected cells. So there's lots of complementary ways, and antibodies do lots of different things. And so if you want to try to figure out which of those quintillion antibodies is the very best, you have to decide what experiment you're going to do in the lab. Which thing are you going to test for? Are you going to ask where it binds? Are you going to ask how broad is it? Meaning, does it bind original coronavirus and alpha, beta, gamma, delta, everything else? How well does it block infection? How well does it prevent, processing of that glycoprotein spike? How well does it inspire your natural killer cells? How well does it inspire this, that, and the other? There's lots of different kinds of experiments you can do and ways you can characterize the antibodies. And the goal in making an antibody for therapy, for either treatment or prevention, is you want to find the very, very best. And the other thing is you might want to make a mixture. You might want to put two together, because if you have one antibody, it hits one footprint. And if the virus makes a mutation there, well, you're out of luck. But if you have two, the virus will not escape that therapy unless it's made two different mutations at the same time to knock them both out. So a cocktail, that mixture, is a pretty safe treatment approach. And also a lot of antibody cocktails and mixtures help each other. Maybe by binding here, it opens up this site. And so the pair together are greater than the sum of the parts, right? They help each other, they're collaborative. So there's lots of different questions to answer. What's the best antibody? What experiment are we going to do to figure it out? How do we build a cocktail? And scientists are experts in different kinds of things. Their expertise might be in figuring out how antibodies signal other immune cells to come. And there are other labs whose expertise is really just in discovering antibodies. And there are other people whose expertise in figuring out how they work in different animal models. And there's no one lab that can really be the best in the world at all these different things at the same time, because they require different background, different expertise. So if the world wants to make the best antibody treatments, we need to draw upon all these different labs and all their different levels of expertise. So we have been able to build global collaborations whereby the expert in how the antibodies work in animals works together with the expert in figuring out what antibodies exist, works together with the experts in figuring out different ways in which they signal to the immune system. So able to combine all those sources of information and get, a better understanding of how the things work. Also, by combining the antibodies discovered by dozens and dozens of labs, we can see how they compare relative to each other. So for example, if you make a panel of antibodies in your lab, you'll have one here and one here. And this one neutralizes Omicron and this one doesn't, but that could just be a one-off thing, right? This antibody works for some reason, this one doesn't. In this international group, we're now comparing 400 different antibodies that people have each trying to advance their therapy. And so we're able to see the reason they neutralize over here, we have a lot like that. It hits a site that doesn't change, that's conserved. Or we find that of all the people raising antibodies against this site, they have this range of behavior. So by looking at many different things and comparing them, you can get a sense of what do antibodies do against the spike rather than what this antibody does or that antibody does. And there's really a place for both. There's a place for scientists that are going to work by themselves and just go as fast as they can to put a therapy in place where there's nothing, because they're racing against a pandemic. And there's a place for a broader approach to compare things and compare techniques and try to get a better understanding of what's happening.
Awesome. I'm going to let you turn to Jen and move on to other topics.
So Jen, I'd like to ask you about T cells. So there are a couple of things. How do antibodies work with T cells? And there's so much in the press about, well, the antibody levels are declining and there's less about the T cells. How do you measure a T cell response? And how do antibodies and T cells work together?
Very good questions. So I think Jim made a comment earlier about how people, scientists are often either splitters or lumpers. And I think in the T cell field, we're immediately going to have to start splitting things down. So when we talk about the T cell response, there are generally two different flavors or categories of T cells that we talk about. One is called CD4 T cells, and one is called CD8 T cells. And I particularly like to study the CD4 T cells, and their job, they're called helper T cells. Their job is to support a lot of different aspects of the immune system. And one of the things that they're particularly good at is supporting the B cell response. So when you receive a vaccine or when you have an infection, the B cells that want to produce these antibodies that we've been talking about need a little bit of help. So these CD4 T cells will come in, they'll say, oh, I recognize this part of the coronavirus or this part of the spike protein, and they'll actually provide contact to the B cells, and they'll help them to produce a really strong antibody response. And so we're interested in studying these cells so that we can understand how we can best support the antibody response, and to understand why some people will make particularly high levels of antibodies or particularly good qualities of antibodies, like you've been talking about. I've already forgotten the second part of your question.
How do you measure a T cell response? How do you know if someone has a good one, a strong one, a medium one?
So there are a lot of different ways to do that. Typically what we do is we would collect a blood sample from someone who's received a vaccine or had an infection, and we will take the white blood cells from their immune system and we'll culture them in the lab, and we'll provide them with little pieces of the virus. So we'll say, okay, well, we'll incubate a little bit of the spike protein with these T cells, and then we look for a reaction from the cells. So maybe they'll start producing some of those soluble proteins that we were talking about before, some of those signaling signals, or maybe they'll start changing some of the markers that they express on their surface. And then we can look for those changes over time, and we can say, okay, this seems to be the number of cells in this person that recognizes this part of the spike protein, or that recognizes this part of the coronavirus. But to be a little bit more sophisticated, we can also try to develop new tools and techniques that allow us to directly identify these cells in someone's blood. And that's been a big focus of our work recently. So we can develop tools that will allow us to directly mark these white blood cells that recognize the spike protein. They make them fluorescent, and then we can measure them in a machine called a flow cytometer. And that allows us to really easily count the number of those cells in the blood, and also to understand what their jobs are, what their roles are, and how they're functioning within the immune system.
So I think you hit on something that explains why so much of the conversation in the media has been about antibody. Like, you know, it's easy to measure the presence and absence of antibody. You can do it in an at-home test. Not everybody has a flow cytometer on their kitchen table. So T cells are a little bit harder to measure. But would you say that one is more important than the other, or does one need both?
I would never say something so controversial as that one is more important than the other. I have many colleagues who are deeply committed to antibodies and the B cell side of the immune system, but it has been a consistent, I guess, issue and at least talking point during the pandemic. A lot of the clinical trials, when we're assessing vaccines, will report the antibody levels that those vaccines elicit. And they can do that in a way that's at least somewhat standardized across different trials. And we can talk about how those antibodies seem to associate with the amount of protection that you get from a vaccine. But measuring T cells it's trickier. People do it in a lot of different ways. And so it becomes a lot more difficult to sort of have a standard idea of what types of T cell responses each of these vaccines are generating. And it's caused a bit more difficulty in trying to understand precisely what role T cells are playing in providing protection or preventing people from having severe disease. And I think it's an issue that's receiving more attention now and a little bit more commentary. And I hope that we can sort of standardize and make it a bit more common to try to study and report on T cells so that we can more clearly identify exactly where they're going to be most important in protecting us from infection or from disease.
And there's a fundamental difference between what an antibody searches for and attaches to versus what a T cell shows or interacts with. Can you talk about the differences there?
Yeah, and I think that's really come into the forefront, especially with Omicron. So we talk a lot about the changes in the Omicron variant and how those changes or mutations can alter the ability of antibodies to recognize the Omicron variant and how it sort of escapes that antibody response from a lot of our vaccines. But T cells are quite different. There are a lot of different portions, both of the spike protein, but also of other parts of the virus that T cells can recognize. And because of that, the ability of T cells to remember the virus isn't quite as affected by all of these mutations that we're seeing across variants. And so it's really raised with Omicron this idea as to whether T cells are going to be able to provide really key memory from our original vaccine responses that will protect us at least from severe disease, and whether that might end up being a bit more effective than some of the antibody responses, either due to mutations or due to antibody responses waning in the blood over time. And so I think there's been some interesting conversations and a good opportunity to try to understand how T cells might protect us, even if we have viruses that do seem to evade the antibody response a little bit.
So T cells will present a short little linear piece of a virus, and it could present a piece of spike, a piece of an internal protein, a piece of some internal enzyme. Does that make it easier for a T cell to cross-react between some common cold coronavirus or this year's coronavirus and next year's coronavirus?
It certainly can. So there was a lot of interest early on where we realized that people who had not yet been exposed to SARS CoV-2 seemed to have some T cell memory responses towards the virus. And that was quite surprising at first because we thought, nobody's seen this virus before. Why would our immune system have any memory or any ability to respond to portions of this? And yes, it seems to be the case that these human coronaviruses, so these are viruses that cause the common cold, and that circulate around the world every year. There are portions of those human coronaviruses, especially in their spike protein, but also in other parts of the virus, that are similar enough to SARS CoV-2 that we have a little bit of a T cell response to those human coronaviruses that also recognizes SARS CoV-2. And there was some really great work done trying to understand whether those immune responses could be beneficial. So did people who have a response and a bit of a memory to those human coronaviruses, did they make a better immune response when they received a vaccine, or were they less likely to get COVID-19? And there has been some evidence suggesting that those T cell responses are helpful, and that they might actually help you produce a slightly better and stronger antibody response actually to a vaccine. So that was a really interesting finding, showing that, all of these viruses, although they can be quite different, our immune systems really are remarkable, and they can find those tiny little pieces that are similar enough that they'll be able to give you that little bit of an advantage if you see a different virus down the line.
So is it enough? Would you rely on that?
I wouldn't rely on it, but I think it's a benefit. It's a benefit. There's also been some questions as to whether it might be a detriment, depending on what parts of those human coronaviruses your immune system recognizes. I think it's always a little bit more complicated than just this is a fantastic thing or this is a horrible thing. But there is some evidence that having that little benefit certainly doesn't hurt.
That has been so fascinating. Thank you so much. It's been so amazing to hear you speak so clearly about your fields of expertise. And I thought perhaps as scientists, shall I switch on my video? This is the message I'm getting. Perhaps you all have in the meantime have thought of ways to ask each other about ways in which to communicate all of these complexities. Again, as we're journalists on the line, I think many of us have learned so much already just from those interactions, but it would be lovely to hear you sort of interrogate each other, if you like, about how to actually communicate this in ways that, everybody can understand. We journalists, and certainly also the audiences we write for. And sometimes, perhaps without the aid of graphics, without the aid of a video, it might have to be on radio, it might be all written word. So go ahead and ask each other about that. Fine.
I have a question for Erica. So a picture's worth 1,000 words. There's been some amazing pictures in this pandemic of the viruses, of antibodies and things like that. And those pictures don't come from outer space. They come from scientists who make data and then make pictures. And it's been an important part of the communication here. I wonder if you could explain to people, because you're sort of a master at this, where do these pictures come from?
Yeah,
what
that's
are they?
my expertise is making those pictures. So we take high resolution images of the pieces of the virus and how they actually interact with the immune system. And these are things that were too small to be seen by any microscope until just a couple of years ago. And, a Nobel Prize a couple of years ago was trying to figure out how to make microscopes with detectors that are sensitive enough to see these things. So the microscopes we use to image these molecules are 11 feet tall, and they send a beam of electrons that's 300,000 volts down there. And so we can actually capture pictures of a couple 100,000 individual molecules of the coronavirus spike anchored onto some human antibody that we're interested in. We average them together and we can make a three-dimensional structure. And it gives us very much the roadmap to understand what we are looking at. Where does this antibody attach? What does it do when it's attaching there? What amino acids are in its footprint so we can understand its vulnerability? How does that coronavirus molecule change its shape? It has pieces that go up and down and move in the course of its function. The antibody can adjust that shape. The coronavirus spike is covered with human sugars, like a wolf in sheep's clothing. And so it's important to understand where those sugars are and what they look like. And we can use those images to actually make better vaccines. So I said a vaccine was like a wanted poster. You want to show a picture of the bad guy. And well, that picture's got to be recent and it's got to be accurate. And, it doesn't help if the bad guy has a photograph with a mustache, a beard, glasses, and a hat if he doesn't anymore. So we can use those images to figure out how to stabilize that molecule to always be in the right shape and structure. So the coronavirus spike is a 3-part molecule and they all come together. And some of the best antibodies we've seen are ones that anchor one part to the other. And so if your vaccine tends to fall apart, you're not going to find those kinds of antibodies. Or the vaccine tends to show surfaces that shouldn't be exposed. You can make antibodies, but antibodies aren't good. So those images help us understand how antibodies work, what sites are vulnerable or not vulnerable to mutagenic escape, and how you might craft a better vaccine. So we're doing all those things at the same time with the complementary sources of information.
Awesome. I wonder if there's a career for a certain type of journalist who would make images. I mean, there must be journalists. I'm not that versed in the field, but can people go to train, not in the science of it, but in the making the images and movies that explain things? Is there a place to learn that? Or?
Yeah, there should be. Because I think if you have the right images that convey scientific accuracy with the simplicity that we need to grasp it, you can get across a lot more meaning in a picture than words, and people are much more likely to look at a picture, especially a moving picture. Mm-hmm. The Global Virus Network used to have a boot camp for journalists to understand virology. It would be great if there was one to also understand how to do scientific illustrations and use that kind of information.
Well, so that's a goal for, an outcome of this call. Could journalists and scientists partner to establish training programs in the graphics? I think they've been very powerful to communicate in this pandemic.
One of the challenges in this pandemic has been how readers are frustrated with the uncertainty. If you want to say, does this work, does it not work? And there's a lot of, well, we don't know. Can y'all talk about some of the reasons why we don't know and where uncertainty comes from? And the way scientists talk to each other, we almost never speak in absolutes, because in biology, there's always some exception. And so we tend to say things like, well, most of the time, this site is resistant to mutation, because there's always an exception. But to convey something in a newspaper article, you need to use generalities.
Mm-hmm.
Yeah, I think that's been, I think that's been a really challenging part of science communication and talking to the media throughout the pandemic, at least for me, because I want to convey things accurately, and I don't want to be misleading. And so I think I agree, as scientists, we never want to say this is 100% what the answer is, or I can absolutely guarantee that this is what's happening, because we know that there are exceptions, there are variables, or we test things in the lab, and there's a bit of variability around that. And so I think it feels like being honest by saying, oh, it's not 100% certain. But then that can be confusing to the public, and that can make it seem like, oh, we don't know what we're talking about, or we don't know what we're saying. And that can get misconstrued as, not being confident in, the concepts that we're putting forward. And I think it's been really important and a bit of a struggle to sort of walk that fine line between giving a clear message and conveying something clearly, while at the same time not promising that it's 100% effective or 100% accurate all of the time.
Yeah, I would have to, I'm just reflecting back on events that happened. I'm thinking about when Tony Fauci, very early on in the pandemic, people said, well, what percentage of people would need to be immune before we could sort of relax? I think he said 75% the first time. And then he came back in a subsequent conversation and said 80 or 85, which, with the confidence that we have in these numbers, really what he was saying was 75 plus or minus 20. I mean, that was sort of what he meant. But then there was a lot of concern in public and in publications whether he was lying, lacking integrity, moving the bar, this sort of thing. So I think we have this impulse to be precise, not to say all or none, to give a number, but sometimes these numbers are not, you might do the experiment and I might do it, and we're going to get different numbers. We're comfortable with that. But I think once a number gets put into the public, it sort of freezes like it's a special artifact. And I don't know how to deal with that. I think that's something that journalists need to explain, that these are estimates that, and they may change, and also if when new data arises, I just read the Einstein biography, Isaacson's Einstein biography. And that seemed one of his magic intellectual powers. He'd get really locked into an idea and try to work that out for about 2 years and then realize, oh, it's wrong. And because new data would come along, and he would abandon the old idea or statement and say, I thought it might be true, but I no longer believe it's true. And I have a new idea. That's a very disconcerting to the public, I think. But that's how science works. It's actually the power of science that we will change our mind when new data contradicts the old idea. And how to communicate, it's not exactly uncertainty. It's an update of the model based on new findings that contradicted the old. And I don't think we articulate that very well.
Yeah, then a good analogy would be like, well, how long does it take to get to the airport? Well, 30 minutes, but today there's traffic, or today the road was closed. And so you could say it's 30 minutes to get to the airport, but on your app, it's going to update that number as traffic changes and as the accident on the road happened and got cleared.
Yeah.
I would love to just jump in here. Thanks, it's Ida. You don't have to put, I'm not going to turn my camera on, but just to say, I'm just so thrilled that you introduced this idea of uncertainty in science, because we certainly find that, it's difficult for journalists, even if they themselves grasp it, to convey that to their audiences. So I was wondering, I love the traffic analogy. I wonder if it is also helpful to think of uncertainty or an evolution of understanding in other areas of science. What brain surgery looked like, 50 years ago is completely different to what it looks like now. Can you think of a few other examples to mention so that journalists can kind of use them and help the public understand this?
Well, another source of variability and uncertainty is, will something work, right? Will this vaccine work? Why did I get infected anyway? And so there's a difference between, there's a concept, the job of the vaccine is to prevent you from getting very sick. You may get infected, but your immune system will clear it and you'll be much better off. It's kind of like, the job of a seatbelt is to make sure you walk away from an accident if you get in one. The seatbelt doesn't prevent you from having an accident. And then it's very hard to talk about absolutes with biology and with populations, because, it depends on, have you had your second booster? How long has it been since you've had that shot? It's kind of like saying, well, will birth control work? Yes, most of the time, but it depends on how you use it. And, there's 2% of the time where it doesn't. So maybe analogies that people can easily understand.
I think maybe we changed a bit how we talked about the idea of, will the vaccines work, throughout the course of the pandemic too. And initially, I think, the WHO indicated that they would like to see vaccines that were more than 50% protective against acquiring infection. And then all of a sudden we had these trials coming out where there was 95% protective efficacy, and that was amazing. And that sort of changed the conversation of how we talked about whether the vaccine would stop you from becoming infected or whether it would stop you from becoming quite seriously sick. And then as time went on, we realized that, over time that protective efficiency was going to decline. We sort of shifted back to talking about protection from severe disease. And so I think sometimes that changing narrative, although it's important, it's the reality of doing science sort of on super speed in the pandemic, created a bit of confusion in the public sphere, and sort of was a bit of a flip-flop in people's minds about what the goals of the vaccines were and what we considered to be a good vaccine or a bad vaccine, and what we wanted to accomplish. And I'm not sure I have an answer to how to do that again next time around, but it certainly did. I think it's the reality of the pandemic and the speed at which science has been progressing.
Absolutely. I think we got to be clear on that. So it's sort of, we have to have an outcome from a vaccine, and then we want to have some stuff. And what are the have-to-haves? The have-to-haves is keep you from dying. You want to keep you out of the hospital. And if you want, bonus points, keep you from having any symptoms at all. And what was, amazing about COVID was some of the early vaccines were so extraordinarily effective across all those boards. We kind of got greedy and we said, oh, it's going to prevent everything. And it did, but that's not sustainable really. And so we should have been emphasizing, you got to keep you alive and ideally keep you out of the hospital. All the other stuff is just bonus points. And I think we were confusing about that. I think we were switching back and forth. The definition of the word protection, maybe, that you brought up. Like, what does protection mean? Protection against severe disease is the core goal of most vaccines.
Thank you so much. I think let's give journalists on the line a chance to ask their questions now. I know some have been posted in the chat. I'm sorry, I didn't mention this. I know somebody posted in the chat, but there is simultaneous translation into French and Spanish here. We perhaps have some questions both in French and Spanish as well. We're gathering them together, but I have also seen some questions in the chat. I think there's a bit of interest in monkeypox, of course, around the world. I mean, journalists are asking the question, should they just shift reporting on this, for example, and so on. I wonder if any of you would like to answer the monkeypox question. The specific question is there a possibility that the outbreaks of monkeypox will to some extent disturb the fight against COVID-19? Are there any plans underway to have the monkeypox vaccine anytime soon? It's in the news, so I'd love to hear just some initial thoughts from any of you. Thank you.
Well, this is one of my pet peeves, so I'm going to chime in. So we've worked on smallpox, monkeypox, cowpox before. We have awesome drugs for this that would be perfectly good to treat and prevent these. Other people have worked on vaccines. So we have all the things in hand. But the next step is to manufacture them safely and cleanly and to do clinical trials and show they're safe. Well, that's about a $25 million to start that process. And the problem is the public, we point at people, companies, the government, but really the public does not have the will to invest proactively against these things. And so we end up with what is called the panic neglect cycle, in which we say, oh my goodness, there's monkeypox. It might take over the world. We should do something about it. And then 2 weeks from now, people say, well, that was in Congo and, it didn't ever get near me. And I don't care. And I flushed that. And literally we've had epidemics, maybe, large epidemics every 12 to 18 months over the last decade. So Zika causing brain damage in, fetuses, chikungunya causing debilitating arthritis. 1 million people in the Caribbean got infected in 1 year, which is very near the US, but all over the world. It was interesting. I was talking to another journalist last week and I mentioned chikungunya and they said, what was that? And I thought, you got to be kidding me. This dominated the news for months, every night on the nightly news, chikungunya. And now people don't even remember it happened. So this, I've got to pivot to monkeypox, and then later I'm going to completely neglect it. We never get the job done for these things, and we're totally reactive. Where what we ought to do is be proactive and say, we know yellow fever is going to cause problems in the world. We know dengue already affects 400 million people a year. We know all these things, and we should invest in them and have them ready before they spread into a place, not wait. For metaphors, I know we're talking metaphors. I sort of think about like, do you want like fires? Do you want to invest in airplanes that will put out enormous regional fires by dropping water or fire retardants? Or would you be better off having a fire extinguisher in your home and you have a little stove fire and you put it out and your house doesn't burn down, your neighborhood doesn't burn down, your city doesn't burn down? So, being ready right at the site, being proactive is so much more efficient, but that's not what we do. We just wait till it happens to us and then we panic. So I think, maintaining attention for longer periods of time would be good for journalists. And I know journalism to some extent is a business, and it's really the problem with the public. The public loses interest, and how to sustain that interest.
Thank you so much. Some of my colleagues have also picked up some of the questions from the floor. Katerina, could I ask you to forward some of the questions you've seen?
Yes, of course, Edith. So I'm just going through here. There's a question from Alphonse Delicana. Is it true that there are some people who can avoid contamination of COVID or AIDS thanks to their natural cells? Perhaps somebody could speak to that question and any thoughts on that concept.
guess
you need prior immunity to ward something off. Jennifer, are you going to say something?
I was just going to talk about the HIV instance, but go ahead.
No, I want to hear about the HIV one.
I was just going to say, it's a little bit more clear cut, I guess, for the case of HIV. So we do know of some individuals who are quite resistant to HIV infection, and that's because of a particular gene that they carry. So there's the receptor that allows HIV to get into our cells and to infect our cells. And there are some individuals that carry a version of that gene that actually makes it so that HIV can't attach to that receptor. And as a result, HIV isn't able to enter the cells in those individuals, and essentially they're quite resistant to HIV infection. So we know that there are some examples where someone's genetics can actually affect their susceptibility to an infectious disease. I think we're not quite clear yet on whether that's the case for SARS CoV-2. There's been a lot of international efforts underway to look at genetic associations with whether people get severe disease or aren't infected, but that isn't quite as clear as some of the examples that we do have, like HIV. But I'll let Erica finish her thought as well.
No, your thought was beautifully conveyed. Just that you need an immune response to ward something off, and there's possible that somebody has some cross-reactive immune response from a similar previous infection that's helped them have a more mild case, or a case where it's asymptomatic, where maybe they did get infected but nobody knew. And apologies, I need to drop off for another meeting, but thank you for including me.
Thanks, Dr. Sapphire. We'll still have Dr. Juno and Dr. Crow on the line. And Dr. Juno, there was a question for you about CD8 cells, just to kind of distinguish again between the different types of CD cells, please.
Yes, so we have our CD4s and our CD8s. So our CD4s are those helper cells that can support other cells in the immune system. CD8s are the killer cells, as Jim was talking about a bit earlier at the start of the session. So CD8 T cells, their job is to look out for pieces of viruses or bacteria or pathogens that might be presented by antigen presenting cells in the immune system. And if they see a piece of something that shouldn't be there in your body, they might say, oh, okay, I recognize that, I'm going to eliminate that cell. And so they actually carry proteins within their cells that are able to destroy these infected cells. And we think that that might be an important mechanism by which we can get rid of cells that have been infected with viruses in this case. And these CD8 T cells basically are sort of the army that goes around in your bloodstream looking for infection and trying to eliminate it.
Could I ask why they're actually called CD4 and 8? Why the numbers 4 and 8? Could you tell us that?
I should know the answer to that.
Well, yeah. So this is one thing I think for journalists, it's really aggravating in that immunology or cells of the immune system do not have branding agencies. And so they start with an internal name sometimes that's not really that helpful. And those names persist and they're not very informative. So the CD system is called cluster of differentiation. That's the C and the D. And it's basically a protein on the surface of cells that we can recognize. And some of the cells have dozens of these. And then as scientists historically were making antibodies that would recognize these cells, they just numbered them 1 through, it's like 300 now. So CD1 was the first one, CD2 was the second one, CD3. So they were arbitrarily numbered using a term called cluster differentiation. And no one would even say that number. You can see immunologists don't use that phrase. We just call it CD because it's such a horrible name. And you can say the same about B and T cells. So B cells were discovered essentially in chickens, and chickens have an organ called a bursa of Fabricius, which humans don't have that. And so the bursa is where the B came from. So we have this chicken in the these cells. They're actually made in the bone marrow of humans. So these cells having to do with the bursa of chicken, which is crazy. You would not do that again, but that's where it came from. So we never say bursa. We don't talk about chickens. We just say B cells. T cells come from an organ that is in humans called the thymus that's in your chest, THYMUS. And so we call them T cells because they're thymus derived or thymus origin cells. But again, we don't go around talking about thymus cells. We talk about T cells. So a lot of the terms in immunology are kind of aggravating. Have to say that.
Yeah, we often get asked about, the different types of T cells that we study, and I study different markers on the surface of cells, and so sometimes they're called CCR6 or CXCR3, which have long versions of those names. They also have different CD numbers, so CD196, and we don't use any of those, and it doesn't mean anything. And the journalists are always saying, okay, what can I say about CCR6? And it's like, no, it's just a term that we use to categorize things effectively. But we like to use as many acronyms as possible to make it quite confusing, I think.
Yeah, and scientists have like naming body. When it gets too out of control, there's naming bodies. And they'll change the names. So you'll remember this virus used to be called, well, it was kind of had China and things like that originally, Wuhan. And then it was NCOV, which Jen was.
Novel coronavirus. It was just novel coronavirus 2019.
And so it had an official name of novel coronavirus 19.
Which just means new.
New, different. And then we changed it again. And immune cells, like even within CD, the ligand of CD40, we used to call CD40 ligand for a very long time. And then people said, oh no, call that CD154. And so even in papers, you see it either way. It's very confusing. So I think, I'm sorry to the journalists that this stuff's so hard.
That's so fascinating. I'd really love to hear from either or both of you, because this question is coming up all the time, and my colleague has asked the question here: why is it that we're seeing lower levels of death and COVID-19 illness and apparently even infection in sub-Saharan Africa? And what about help us actually understand that from the perspective of the immune system, if that is part of the explanation, in your view?
I guess I'm not going to pretend to be an epidemiological expert on the South African viruses, but I think South Africa has had a bit of an interesting experience with different SARS CoV-2 variants. So of course there was a wave of the beta variant a year ago or so now, which spread more throughout South Africa than a lot of other parts of the world. And then more recently, there was that really large wave of the BA1 Omicron variant. And I think we're starting to see now, as these different variants of Omicron, so we talk about BA1, BA2, more recently BA4 and BA5, that there seem to be patterns across some countries. So places where there's been a really large wave of BA2 infections, we maybe aren't seeing quite as much BA4 and BA5 coming in and taking off. And these patterns might be related to the immune response to the different variants. And this might be related to, and Jim can probably speak to this more, about how well antibodies are able to recognize one variant versus another variant, depending on the viruses that have sort of moved through a population. And so I think South Africa has had a bit of a unique situation compared to some other countries globally, and that might be contributing to why they're seeing slightly different patterns of disease.
And could I actually ask specifically about the rest of sub-Saharan Africa? I mean, in countries in West Africa, we continue to hear reports of seemingly really low levels of infection and certainly of death due to COVID-19. Any idea?
I think in general, vaccines have changed the profile of the relative amounts of death and severe disease that we're going to see. But I must admit, I'm unfortunately not quite as familiar with some of the infection patterns of other countries in sub-Saharan Africa, which is certainly my lack of knowledge, I'm sorry to say.
Well, I don't know all the answers here either, but I think it's important to think of this in two ways. One is the difference in the numbers real? So if you have a country, and I'm not talking about Africa specifically here, if you have a country for any disease and they don't do any testing, then their incidence and prevalence will be zero. And so you can say, why is the infection so low? It's because they don't test. Now, sub-Saharan Africa has actually been really interesting during pandemic because there's places where testing was not available, so the numbers were not available. You really could not do the epidemiology, and yet the scientific prowess of certain institutions in South Africa, because they're leading the way on HIV vaccine and testing and treatment and so on, there was actually the highest level of capacity to do real-time high throughput testing in the world. And so you sort of had this perception outbreaks are exploding out of South Africa, when in fact it may have been that the South Africans were the first to know, because their testing capabilities were so high, and they have a lot of sequencing capability. So they not only knew they had lots of COVID, but they knew which variants, and they could sort through thousands at a time and say, we have a new variant. So that's kind of on the, are the numbers high in South Africa, or do they just know about it more? And some of the other countries, are they low, or do they just didn't test? Now, I do think it's a fair question. Are there instances where an entire population is lower risk for some reasons? And there are examples where, in particularly in Africa, it's been talked about that the genetics are different in certain areas of the world, and certainly there are genetic what are called polymorphisms, differences between human genomes, and it may be that certain African populations are resistant to certain infections, vis-a-vis other populations. Another idea in that idea is not genetic, but has to do with exposures and immunity. So there's a whole field called the Hygiene Hypothesis, in which kids that play outside when they're little in the dirt get exposed to a lot of environmental things. And actually that's to their benefit usually. So they get lots of teaching of their immune system, whereas you have other cultures where the kids don't play outside, they're been obsessively clean, almost like sterile environments. They don't have that education early on, and they often end up with allergy. So you see a lot of allergy in super clean childhood environments, and that's not good, actually. That's a bad outcome. And so the Hygiene Hypothesis has posited that many areas, including sub-Saharan Africa, have a better education of the immune system. They also have things like malaria or other parasites are more prevalent. And you may, you don't want to get malaria, but if you've had various other parasitic infections, that may give you some more general resilience in your immune system. So I don't know the answers, but I think there's a lot of knobs you could turn around why certain areas have different numbers. It's pretty complex itself.
Thanks for that, Dr. Crow. I think it's just a reminder to us that, how new, how novel this virus still is, how much there is still to discover. I mean, we're still discovering a lot about Ebola and HIV, etc. And, for journalists to keep on looking out for asking those questions and looking out for folks who study it very specifically. But thank you so much for your responses. I actually wanted to just quickly follow up on the Hygiene Hypothesis and ask you, it sometimes sounds to us when we follow narratives in the media or on WhatsApp groups, in community discussions amongst neighbors or whatever the case may be, that Hygiene Hypothesis is sometimes morphed into the idea that one's natural immunity will take care of a pathogen. And I know you spoke about it right at the beginning, but could you just, because I am thinking it's such an important concept to get right, just to hear what, from both of you, what the kind of most adequate, accurate response would be. If one hears somebody saying, hold on, I'll just boost my own immune system, I'll take vitamin D, I'll eat vegetables, because that narrative is out there. And we'd love to hear from you what the appropriate response to that is.
Well, I think the human immune system is amazing and has extraordinary capabilities to deal with stuff. So I would not denigrate the capacity of the human immune system. We can make responses to anything that comes at us. In fact, you can make a synthetic chemical that never existed on planet Earth and inject it into people, and they'll make a response to it. So the information and the capacity in the immune system is extraordinary. And there are a lot of viruses and bacteria that generally don't kill you. So if you get the common cold like rhinovirus, you may have some symptoms and you have a common cold and you get over it, and your immune system has been educated by that and will protect you in the future. So that's fine. But you don't want to do that with fatal diseases, right? So if a disease has the capacity to fail, to cause fatality, then it can overcome the level of natural immunity that we have, the natural resistance. So there are many potentially fatal infections, and you see HIV is invariably fatal if you get it. Rabies is always fatal when you get it, unless you have just extraordinary treatment, et cetera. So, and COVID falls in this middle ground where many people do die, and you don't want to trust a system that is not built for fatal viruses to save you. Get lucky. Lots of people get lucky. It's not, the number is, I would say, moderate in terms of its lethality. But if you're the one dying, it was a bad choice. And a lot of people have died. We have millions of people that died of COVID. So relying on sort of the luck of what might happen with natural immunity is a bad choice for COVID. It's an okay choice for the common cold. So I think it sort of depends on the germ you're talking about.
And I have to say, I don't love the phrasing natural immunity, because I think it implies that we have a form of immunity that's unnatural, or that the vaccines are producing an immune response that is not correct or normal in some way. And the key feature of the adaptive immune response is that the first time that you see something like a new virus, for example, your immune system is designed to remember it, so that the next time you're exposed to it, the immune system can react more quickly and more strongly and do effectively a better job at trying to get rid of that virus. And really, with vaccines, we're just trying to accelerate that process in a safer way. So we're just showing what we think is a really important part of the virus to your immune system in advance, so that it can develop that memory and respond more effectively the next time that you see it. And so I think, again, this idea that we have natural immunity or versus fake immunity or synthetic immunity or something is not, I don't love that phrasing, because I think it implies that the vaccines are doing something, or teaching our immune system in a different way, or something that we wouldn't want. And I think that's doesn't do anyone a lot of favors.
Thank you.
I think that's fair to say. Immunologists don't use that term, actually.
Right.
We don't use that term, yeah.
Thank you. So talking about terms and expressions, my colleague has posted in the chat a glossary that we've put together of various terms used in talking about immunity. So that's obviously for journalists on the call to use. We'll also mail it to everybody who's attending, in case anyone's no longer here or can't see it in the chat. So I hope you enjoy that. I wanted to ask a quick question, just talking about and I see there's a really fascinating question about, let's go to the question in the chat first, actually. But I did want to ask Dr. Crowe something about antibodies. The question in the chat is that we know that SARS CoV-2 can, in severe cases, elicit a cytokine storm, a kind of overreaction, in other words, of the immune system. So in that case, how do you approach the dosing of vaccines or antibody treatments? And my question to Dr. Crowe is about antibody treatments, but let's handle this one first in a way that doesn't, increase the risk of this happening or risk this happening. Is that related to dosage? Please help us unpack that.
Well, in terms of treatment, there's several things you can do. One is, if you have a drug that actually inhibits the virus itself, less virus usually is good. So antibodies can do that. There are also what you would typically think of drugs. They're chemicals. So people call those small molecule drugs. And there are now some of these that are licensed that you could take by pill or IV. So those are the two sort of treatments. These treatments only work well early during infection. They either prevent or treat. The more and more virus you have, the harder it is to treat, which is later in the infection. By the time you're in the hospital, it may be that the virus is actually going away because of the immune system. And now the disease you have is due to your immune system. Your immune system's coming in. It's like a war zone. It's not only putting antibodies, but it's killing infected cells. And that's a double-edged sword. You're eliminating the virus factories, but you're also killing your own cells. And temporarily, that's a bargain that you'll take, but you don't want to do that too long. And if that fire that's burning, if that gets out of hand, you'll die. There'll be too much inflammation, too much virus, and you're just killing yourself, basically. And so a third way that people have thought about treating the disease, not the virus, but the disease, is try to tamp things down to reduce the immune system response. So drugs have been used like steroids. So steroids are anti-inflammatory drugs. So the idea is if you reduce the inflammation, the person won't be as sick. You know, the risk is then the virus will come back. So the immune system's there to kill the virus, but it's also killing you, and you have to sort of play with the anti-inflammatories. And there's other anti-inflammatories that have been used. So I think the longer you wait in the disease, the harder it is to deal with things. And at the end stage, the immune system is not just helping you, it's also killing you.
Mm-hmm. Dr. Crow, just a follow-up. I know that Dr. Sapphire had said to me yesterday that there's a question she'd really love to ask you, but I know she's not here, so I'll ask on her behalf. Why not, I mean, especially since antibodies are your field of expertise and great interest, why aren't more people demanding taking antibodies when they're sick with COVID? Why do you think it is so?
Well, antibodies are very interesting as a technology. We started using them in the late 1800s for rabies and diphtheria and tetanus. And the way it was done originally was to immunize or infect a horse and collect horse serum, the antibodies from horses, and give that to people. And that turned out to be pretty effective, actually. And the first Nobel Prize was given for what were called antitoxins, which later became the word antibody. And so that was, at the beginning of modern science, people were using antibodies for infectious disease. But then when monoclonal antibodies, this drug technology that we've been talking about, when that was invented in the late 1970s, it got shifted to a focus on cancer and autoimmune diseases. And so there are over 100 drugs that are antibodies that are licensed now, but they're just used for cancer and autoimmunity. If you talk to a cancer doctor, they say, oh, I love antibodies. I use them for all sorts of cancers. But we haven't been using them for infectious disease. Prior to COVID, really, there was only, 1, 2, 3 sort of antibodies, one for a pneumonia virus called RSV, one set for Ebola, and so on. So infectious disease doctors, oddly, are not used to using them. Primary care doctors are not used to using them. They don't know how to use them when. They're just less familiar. And so I think that the medical community doesn't know that much about them. Patients don't know about them. They don't know what they are. They don't know to ask for them and when. So I think that's been the biggest barrier, the lack of familiarity. And then finally, the cost is relatively high compared to a vaccine. So an antibody might cost hundreds or thousands, whereas vaccine costs, I don't know the cost exactly of the COVID ones, but $50 to $300 or something like that. So they're a little offset in price, and that's just sort of a supply and demand. I think if we used antibodies more, the price would drop, but they're relatively expensive, and this has just been sort of a beta test of antibodies. COVID was a beta test. We tried them out. They work. We learned a lot of things. Another thing I didn't mention is we learned if you have to give it IV by an infusion, hanging with a bag of liquid for an hour, that's kind of a losing strategy. It's really hard to do that in lots of people. So the antibodies are going to have to be given by shot, just like a vaccine. And actually the ones that we developed for COVID, that's how they're given. And it's so much easier. You just shoot people up and they're good, rather than they don't have to sit there for 2 hours. So we've learned a lot during COVID about how to use antibodies. And I think they'll be a much bigger part of the future of managing infectious diseases.
Absolutely incredible, fascinating. Thank you. There's more than one question about herd immunity or population immunity. Perhaps in the broadest way, can I ask, why does that percentage seem to differ from one pathogen to the other? Just help us understand that. Or the target percentage, I should say.
I guess broadly, there are a lot of factors that will influence the transmission of a pathogen within a population. So that can depend on the mode of transmission, first of all. So as we've seen, the mode of transmission seems to be a bit different for the monkeypox versus COVID. And so that's going to influence how a pathogen spreads throughout a population. Then there are going to be differences in, as we've seen with SARS CoV-2, how infectious are different variants. So we've seen, real changes in how infectious, the original strain of coronavirus was compared to what we're seeing with changes now in Omicron. There's also going to be different levels of immunity across a population. There'll be different proportions of individuals who are protected from infection versus protected from severe disease. And so all of those things are going to come together to make a complex situation, I think, when it comes to really modeling how a pathogen is going to spread throughout a population or a group.
That's wonderful. I see such an important question here. I know we have only 5 minutes left, and I'm just going to read it out saying, if there's one key point you could highlight about the importance of vaccines to boost our immunity and protect communities, what would that be? And particularly in the current context of misinformation, anti-vaccination sentiments, etc. What would be that one message that we should all, hear from you?
Vaccines are good and you should get them. That's the bottom line. I think, one thing we haven't talked about today is the idea of risk and people's tolerance for risk, fear, and so on. If you look at the data on the vaccines that we have for COVID, the risk of side effects or what are called adverse events is extraordinarily low. And the benefit is very high. So the overwhelming odds are that your immune system is going to boost up to a higher protective level, and you will be protected against bad things happening to you, like going to the hospital or dying. And the odds of you having a side effect are infinitesimally small. So why would you not do that? And there are political reasons and social reasons that have nothing to do with a logical, data-driven thing. But I'm just saying we should clearly not beat around the bush and say, these vaccines are awesome, they have benefit, they're safe, and you should get them. That's what we need to tell people.
And I would add, get your third dose. That makes a real difference to your antibody response, especially against some of the variants. And I think that's an important message as well. Stay up to date with your third dose.
Thank you so much. What a wonderful message to close on. I think that's it. Thank you so much for your time. Thank you so much, everybody, for joining. As I mentioned earlier, we will make the video available. It can be, of course, made available also in all 3 languages, English, Spanish, and French. And we are also going to produce sort of shorter versions. So for instance, there were discussions on population immunity, there were discussions on antibodies, on T cells, and so on, around specific themes, which I hope you'll find useful for your stories around these specific areas. So thank you so much to Dr. Sapphire, Professor Sapphire in her absence, to you, Dr. Juno. Again, it is now 3:30 in the morning in Melbourne. You are amazing. You're clearly an all-night person. So really thank you for that. Thank you also, Dr. Crow, so much for your insights. Thanks to both of you. Thanks to everybody on the line. I think we at Internews would definitely want to explore this topic further. So we would like to hear more from you. Thanks again, of course, to our colleagues, wonderful supporters at the Sabin Vaccine Institute. Thank you to all my colleagues who've helped with all of the elements of putting this together. And thank you all journalists for your interest in this, for your ongoing interest in this very important topic. Goodbye.